Hello, and welcome to Completely and Utterly Sporing, a podcast about bacterial endospore formation, where it shouldn't matter if those words mean absolutely nothing to you currently. I'm Kit, a biomedical science student and a spore enthusiast. And this is Callum.
Hello. Yes, I am the person who is going to be who knows absolutely nothing about spores. So you can teach me about this.
Astrophysics student who does not care about biology but does like asking questions.
Indeed.
Over this series, we are going to hopefully take you through the process of spore formation and what the concept of commitment means in that context, so that I get to talk about techniques and findings and implications of an undergraduate summer project that I have just completed on the topic. He should hopefully be there to stop me when I forget people don't care about biology if they don't actively study it, and the people might not know what things are. So hopefully I should be able to explain things in a way that is clear to a full audience of people, rather than just people that are already invested in spores.
I think for the viewers, they have that option of they have the option if they do want to stop you, they can just pause and then exit Spotify or YouTube, whichever one you're watching this on. But hopefully I'll be able to before then stop you and ask a question because it is generally a very interesting topic if you can get through all of the biology jargon which usually surrounds these kind of topics.
Yeah, hopefully, because Callum will be here every episode, he should gain knowledge at the same rate as a listener does. So hopefully his questions will stay relevant to the kind of level of understanding that you have as you go along.
But yeah, first off, let's hear about what the main content of this is, which is spores, because it's not a very familiar, unlike things like bacteria and viruses, which I guess you normally hear about. Spores are not something that I suppose is colloquially known about. So what are spores?
Yeah. So this episode is all about spores and how they form. And you said that they weren't bacteria. Spores are bacteria. Endospores.
Oh, okay, right.
Bacterial endospores. No, sorry. When people talk about spores, they often think of fungi and the idea of having like spores in the air that is generally how fungi reproduce, but these are not the same kind of spores. Bacteria use spores as a dormancy, so a number of bacteria, not all bacteria, can, if there is a lack of food, or they get a bit too hot, or they're a bit dry, or there's kind of a harsh environment, it triggers a number of survival responses, and the kind of last ditch attempt to not die for these kinds of bacteria is to form a spore. So they kind of so
a spore is not really it's a kind of separate entity to bacteria. It's more like a survival mechanism essentially for a bacteria if it enters conditions which are not favorable, is what you're saying.
Yeah, so it's a cell differentiation process. So you know how like stem cells turn into other things.
They differentiate
by making choices.
Yeah.
So they make choices. This is similar because it's a set of choices, but then the end point is dormancy. So when they enter spore, like the spore form, they are metabolically inactive. So they kind of protect their DNA, but they're not reproducing, they're not replenishing anything. It just sort of freezes in a state of, I'll start growing again when there's food.
How long can it actually stay in this dormancy period? Because obviously the bacteria will obviously need resources to, I suppose, continue living, or at least that would be my idea of it. But then this is obviously a state of dormancy where there perhaps isn't any food available. So is there any limit to how long it can actually stay in this form, essentially?
It'll depend on the species that is kind of doing it, and also the health of the spore itself. So when cells enter sporulation, that's a set of steps that allows it to form this dormant structure, and depending on how well it does that, it may or may not be fully able to maintain it, but it should hypothetically be able to maintain it for a very, very long time, because of the fact that it doesn't need food or water. It's just complete, because it's not doing anything inside it. It's just sort of holding information. it doesn't need anything. So like it doesn't need to breathe. It doesn't need to, or well, not breathe, but respire or undergo any like processes, because it's not replenishing its proteins or you doing any sort of energetic processes. It's a highly energetic process to form the spore, but once it's done that, it's kind of just stops until it can detect stuff.
So frozen is quite a good word for it, really, I suppose, because it is
Yeah.
like all of the processes of it stop, and that's why it doesn't need any resources.
Yeah. So yeah, people think some of the early kind of legends about like Egyptian curses and things like that are potentially the fact that people would open these kind of coffins and stuff, or like sarcophaguses, and they'd get infected by spores that were suddenly exposed to oxygen, or other, not just spores, other types of sort of maybe viruses or bacteria that were on ancient remains and things. But spores are a pretty good example of the sort of thing that would do something like that. It's just that actually they can kind of, the whole concept of sporulation is that they can survive indefinitely until they've got the correct conditions where they can detect that. And then there's another set of differentiation processes where it's called germination, like with seeds. So it comes out and it just goes back into normal division like a perfectly healthy cell, because it's still got all the information to act like a healthy cell. It's just not using it.
Obviously, the dormancy period, I suppose, is maybe not too difficult to understand as everything's frozen. But I suppose the thing that's maybe a bit more interesting to me is this ability to transfer into and out of this dormancy period, especially when, as you mentioned, with the example of the Egyptian curses, when it's suddenly exposed to oxygen or something. So it must have some sort of like sensors or receptors in a way on the spore that allows it to know when there is favorable conditions. So is that something that it does have?
Any bacterial cells have the ability, they've got, because of their membranes, they can detect, they release chemicals and chemicals travel through their membranes, and that can kind of activate decision pathways. To put it in kind of easier terms, it's like a flow chart almost.
Yeah, it's like what you see with computer algorithms, essentially.
Yes,
got,
exactly.
If this happens, then do this. If not, then stay in dormancy period, essentially.
Yes. Yeah. Well, the decision to enter dormancy is similar to that, because they do when I'm talking about spores, I'm going to mainly be talking about a species called Bacillus subtilis, because it's the one that we've basically science in general has decided that's the easiest one to study, because it's harmless and really easy to manipulate. So we just use that. So
okay.
For example, that one, it can, if it's starting to get a bit crowded in a space, or it's a little bit hungry, rather there's not like enough nutrients to go around, it can do, its first detection will be how many of us are there. So the cells, though they're individual cells, and bacteria are obviously single-celled organisms that are completely self-sustaining by themselves, they can communicate with the other cells of the same species in a community to detect how many of them they are, and that can allow them to regulate their response to the environment. So if they were maybe a little bit hungry, if there was a little bit of kind of scarcity of nutrients, they can form something called a biofilm, which is where they kind of release proteins to make like a sticky matrix, and then they can all stick together. And that helps survival, because they can kind of share resources. And then that's like the first attempt is sharing resources, and then the second attempt is, right, there's not enough food for that many of us at all, we can't form a biofilm, we go straight to spores. So some of them will just be kind of martyred almost, where they decide to die and release their nutrients, and then those nutrients, because it's a really,
of the group,
yes, because though they are single-celled organisms, they are communities, they are colonies of single-celled organisms. So they do, their main objective is survival and reproduction. So despite it's kind of the good of the population. So they do have the ability to kind of sacrifice themselves, release nutrients that then the other cells can use to have enough energy to complete sporulation, because though once you get there, really nice and easy to survive, the process itself involves turning on and off.
Takes a lot of energy, yeah.
Yes, so many genes and proteins. You're basically entering a whole other metabolism and switching. We will get to this hopefully later in the episode, but a whole number of things need to happen for something to get from a normal cell to a spore. Because it's a whole like a cascade of events to put the DNA in the right place, to put the coat in the right place, to make sure that they are able to resist all of the things that they need to be able to survive through. It's a massive, massive change. And all of those changes do take energy, because you've got to move things around and make things and put things places and things like that. So it's a lot of
work.
It's a lot of work. And why would they do that? If all of them form, the concept is, if something forms a spore, once it gets to the correct conditions to survive, it can come out and it can start reproducing. So it'll just repopulate an entire, hypothetically, if the nutrients are good, it can go back to its really quite fast reproduction, and it can form colonies overnight. So you don't need all of them to form spores. You just need some healthy spores so that they can then germinate and successfully repopulate once it's okay. You don't need an entire colony.
Yeah.
No, there are plenty that won't form spores and they'll just die. And then that doesn't really affect the number, because you've got some successful spores. Once they've got food, they'll just start reproducing. They're good at it.
I suppose any time in this context, especially when you're doing it in the lab, you're talking about like potentially like thousands of these things, and so it's not really if one or two of them don't end up forming spores. And presumably, you mentioned that some of them might be sacrificed, to use a particular term, might be sacrificed for the good of the group when forming this when entering the and becoming spores. And presumably that's the same on the other end as well. If it's coming out of this stage, would you also need to, I guess, sacrifice a few to have enough energy to actually, again, go back to the initial metabolism state, as you so put it?
No, because
okay.
the whole concept. No, that's. I mean, it makes sense hypothetically, but no, because of the fact that the only reason that they would germinate is if there's enough nutrients to do so, because that's what allows them to get out of their dormant state.
Oh, of course, yeah.
So there's no reason why they would need to, because the whole point is that there's enough. If they can detect there's enough food, they won't come out of dormancy unless there's food. Or if we often give the example of nutrients, but also it could be it's too dry, and when there's enough like moisture again in the environment, they can come out of. Or it's a bit warm, and then when it gets cold enough again, they can come out of it. But it's just they can detect when something is not safe anymore, and then they can go in, and then when the conditions are safe again, they can come out. But because of the fact that this is such a complicated process, and because it is so energetically expensive, is the word that how we describe it, it does require a degree of certainty, and it has to be kind of, you have to decide, yes, I am going to form a spore, and then you form a spore, and then when I can detect that it is safe again, I will come back out. If you think, oh, I'm a bit hungry, I'm going to form a spore, and then there was some food halfway through, you'd still have to finish forming a spore, because the process is that complex that you can't just turn around halfway through.
Yeah, see, this is, I think we've definitely talked about this before, because for people listening, the one viewer, for Kit and I have talked about this a lot before, and essentially, obviously we'll get to this, but the point of your project was to, I guess, intervene halfway through this process of forming a spore and as you quite as you said then, it's a decision. If the bacteria decides that it's forming a spore, it is committed to that in a way until
Yes.
until it enters that spore phase, and then once it is has entered that spore phase, it then won't come out of that until it detects that the conditions are I guess good enough again to come out of that dormancy phase and become I guess what we think of as a standard bacteria again. So I guess the one question I had, I guess before we might move on to talking a little bit about that, is how long does this process actually take to reach the dormancy phase once it's, I guess, quote unquote, decided to enter the spore phase? Because it must take a reasonable amount of time for you to actually be able to intervene. It's not just like something that happens in mere seconds, presumably. I don't know.
No, they are, again, we use Bacillus because of the fact that it's really quite easy to use. It's nice and quick. It's good at responding to things. We can mess with it very easily, first of all, and also it'll respond to things fairly quickly. Other people working on harder organisms to manipulate have much longer tasks than we do. But I guess for us, there are different stages of sporulation, which I will hopefully go into in a minute. But depending, due to the way that we were simulating things in a lab, we were interested in just the first few steps of sporulation because we want to know the point at which it can't turn around anymore, the idea of commitment. But the full process takes hours, not days,
Oh wow,
hours.
okay.
So no,
actually longer than I was expecting them, to be fair.
Yeah, I was working because of the fact that we were trying to look at just those first few steps. We were working with mutants, so they were kind of forced to stop after maybe they weren't allowed to complete step 3, basically. So we were growing, but those were growing for 2.5, 3 hours, and they were getting stuck. But by the time that it had been 2 or 3 hours, all of the ones that were going to form spores were stuck at step 3. But I know that I worked with a PhD student, and her and the rest of the people in the lab that do work with fully healthy spores at other times, the way that you will check what stage they're in would be to take a sample and put them under the microscope. And they often check things after 2.5 hours as a spore. And then you might look 1 and 2 hours after that. Once you get to, sorry, when I say as a spore, I mean as the step 3 that we got stuck to, because that's what I got used to spores being. But obviously they are mutants, so they are not able to continue. But I know that for taking pictures, people would often use T7 or 7 hours after their initial introduction to sporulation media, is what we call it, the basically just salty water that doesn't have food in it. And it would be 7 hours after their exposure or 7 hours after their re-transfer to different...
The salty water? Or the different conditions?
So depending on because of the fact that a lot of both my investigation and a lot of the other things in the lab were to do with those first few steps, sometimes you would put them in the salty water for a few hours and then take them back into lots of nutrients to see whether they could turn around or not, because we're trying to get them to turn around to understand what helps them turn around. But I know that, yes, about 7 hours you can take pictures and you can see the fully formed cortex, which is the kind of resistant layers on the outside.
So essentially 7 hours, let's say, that's the time between the bacteria as it was, I guess, for initially entering these I guess harsh conditions, or these unfavorable conditions, I suppose you would say, and then it's the 7 hours to complete the full process. But then I suppose what we're going to talk about now, and what we're going to talk about for a lot more of this podcast, is that there are certain stages that this bacteria has to go through before it forms a fully formed spore that you can see. This is the thing that takes 7 hours, and you are obviously intervening with that process halfway through, as you said, step 3 out of, I'm not sure how many as such, but so like somewhere, I guess, because it's 2.5 hours until you intervene. So you kind of change the conditions halfway through the sporulation process, and that's what you were really doing for most of your time doing your experiments, pretty much.
Yeah, we worked with a mutant that had kind of been forced to stop, and then we put it through different conditions to see what it would do when forced to stop, what it would do to get out of that corner that we'd kind of put it in.
Yeah, can I just check? So you say a mutant. So this is something that so the one that you're using isn't, am I correct in saying, is not anything to do with the conditions it's in? It's because it's a mutant, so its genes are actually different from what you would have as a standard bacteria, and because of that mutation, it stops the sporulation process halfway through, or doesn't reach the final stage, but it stops after, well, as you said, like about 2.5 hours, correct?
Yes, so the way that sporulation works, you've got a cascade of events, and if you delete something, because we can do that, which is still absolutely amazing to me. It's just very cool.
Absolutely. No, the fact
Yeah.
that you can delete and change certain genes is no, you're right. It's really incredible.
Yeah.
And that is
So
something you were able to do.
Yeah, so it's like removing, the way I've described it before is like a series of extension leads, and you turn it on at the wall, and then you've got that maybe an extension lead is then plugged into a new extension lead, because you've got kind of sections of steps that can happen. Like there's you might have like 100 genes that control getting from step 2 to step 3, and then when step 3 is on, then that's another 200 genes that then help with the next step, and it's like a cascade where you've got almost sections of the genome being turned on, which means that if you delete it, almost like unplugging an extension lead, you've stopped the flow of things happening, so it can only get to the end of step. You're not turning on the things that you need for the next series of changes.
Yeah,
so
you've removed those genes that are needed to continue the process of sporulation, essentially.
Yes.
And because you've removed them, it's limited at step 2.
Yeah, well, it's slightly, because of the fact that it's so many different genes are involved in each step, the way that we can work to make it slightly more streamlined is you can delete the thing that turns the genes on for that step. So you're technically knocking out the activity of 200 genes, but you're just removing one gene, because once that gene turns on, you're letting 200 turn on. So it's much easier to just go, just stop that one, and then none of the other ones will do anything. Yeah.
No, that does make sense. We should say, do not try this at home where you actually plug multiple extension leads in, because you might start a fire. But it's a very good analogy that you need these certain ones to make, even though there's hundreds of genes involved in the process, you have identified this one gene, which is absolutely necessary to turn all of the other genes on. And that means that you are fairly, not, I wouldn't say easily, because it's not easy, but you are able to, straightforward, I suppose, is a better way of putting it, stop this sporulation process. And so does that mean when you're cutting or stopping it at this stage 3, I suppose, as we're saying, does it then just stay as it is? If, say, you were to say the conditions remained exactly the same for these mutants, would it just stay as it is, or would it start to reverse in a way?
Well, that is the concept of the research, because if you stop something at step 3, let's call it step, I will get to it, but at the moment we're calling it step 3. We stop something at step 3. So if it's left in salty water, and this mutant, it can't continue with the process to form a complete spore, it undergoes lysis, which just means it breaks up and dies.
Yeah, no, that makes sense.
But if you transfer it to sugary water, which we're going to call that's what we can call it, because that's what it is. It's a nutrient-rich media which contains a lot of different sugars and some amino acids and some salts that the that are needed for survival, like magnesium.
Basically, some really nice conditions for the bacteria. After you've tortured it to put it in this sporulation state, it's very nice conditions for the bacteria. That,
yes, we put it in like,
from what you're suggesting, might give it a reason to turn back.
Yes, hypothetically, because that we're basically putting it in the conditions that it would be able to detect as a spore and germinate. So to come out, it's the sort of conditions that it would come out of sporulation for.
Yes.
But it's stuck partway through. Now, what we've done is deleted the step. So we know that the turning around point is between step 2 and step 3. So if you delete step 3,
okay,
it can't go further, but it shouldn't be able to turn around by itself because it's done step 2. So when step 2 turns on, it turns on 100 genes, and some of those 100 genes stop it from turning around. So if we put it in a condition where it has to have step 2, but can't have past step 3, it should hopefully mutate itself to try and survive, because of the fact that mutations happen in bacteria pretty easily, and survival of the fittest, you've got the fact that evolution of populations will occur, because if a mutation helps something turn around, it'll survive and nothing else will. So hopefully then we can form a population that's able to turn around. And if we know what is responsible for the turning around, then we know what is responsible for it not being able to turn around, because that's just where the change is.
Okay, so hang on. I think I'm going to need to just
Sorry,
clarify.
That was quite a lot.
No, it's fine. No, it's fine. But no, so we can clarify this. So what I think you're saying is that it stops at stage 3, or that's where these mutants stop, and then you're putting it in favorable conditions. Now, there's obviously some genes that are turned on during stage 2, so the previous step before it's shortcuts essentially or stops, that I guess is this concept of commitment, essentially, where it's committed to becoming a spore. And so am I right in saying that because those genes are turned on and it can't turn around at this stage, you're in a way forcing the bacteria to mutate so that it can turn around again? Is that correct?
Yes. Yeah. And the reason that's exactly it. The reason why we're doing this is because of the amazing molecular biology advances that we've had in the past 20, 50 years. We can sequence things, we can find out entire genomes, we can compare a mutant to an average wild type, is what we call it, when it hasn't mutated. So like a normal bacteria.
Your average Joe bacteria, yes.
Yes, average Joe bacteria. And you can compare and you can see where the changes are. And if you know, right, so maybe there's a protein, one of the genes at position 54 on the genome, there is a change, and the only change in function in the bacteria is now it can turn around. You know that that gene, position 54, was what's stopping it from turning around.
Yeah, no, that makes sense.
And yeah, so
you're just looking at before and after cases essentially, and seeing what's the behavioral difference that is associated with that essentially.
Exactly. Yeah. So we're trying. There's, because of the fact that we use bacillus, we use a nice, easy organism. It's been really well characterized. There are database lists of absolutely everything and where it's kept. We don't know the function of absolutely all the genes, but we have names for them. So you can see where they're supposed to be, and you can think, and you can maybe identify that the function of it is to stop commitment or to cause commitment. And if it's deleted, that stops it. But we understand a lot about this bacteria, so it's very useful to work with, because it's really easy to know when something's changed, because you can get someone to tell you. Yes, you can send it to someone that will read the code for you and come back and say, well, at 27 and 54, there are 2 different proteins, or it makes 2 different proteins now.
Yeah. And I guess that was going to be my next thought when you said that, because we say that obviously when people say that the genome has been, I guess, fully categorized, I can't remember the exact word that you used. It doesn't necessarily mean that we know what all of those genes do and what they're for by any stretch of the imagination, especially because there must be thousands of genes, or like, I don't know the proper scaling for it, but there's so many of these, and we don't know fully what every single gene does and what its purpose is. And this is essentially just looking at one case, one particular, or maybe just a few genes, for example, and identifying their purpose by, as we've already described, comparing the genomes of the two different mutants in a way.
Yeah. At the moment, we found out people are finding out what different genes do still, and there are a lot that have been worked out, but because it's such a vast, well, any organism has countless genes, and it's very hard to work out the function because some of them are stabilizing things or helping something do something, or you know how in maybe GCSE biology you do your Punnett squares and you find out that if your mum has blue eyes and your dad has brown
I
eyes,
remember these.
you work out the probability that someone will have blue eyes or brown eyes. That is
dominant and recessive alleles. I remember that phrase.
Yes. So that is obviously bacteria asexually reproduce, so you don't have the concept of Punnett squares or anything like that. But the concept there of the fact that a single gene is deciding what color eyes you have is false. There are some characteristics that are decided by a single gene, but in most cases, it's the kind of cooperation of multiple genes at multiple sites on the genome. So you might have something at position 36 and something at 270. You might have like 10 different genes that contribute to your genetic disposition for height and things like that. So you've got.
I wish I could change that for mine, to be honest. But yeah. No, but you're.
Yeah.
No, but it's like, no, but I get, yeah. So it's once again another one of those cases where you get to like A level and you say, oh well, everything we taught you at GCSE is actually wrong in a way, and then you get to university and they say, oh well, everything we actually taught you at A level is wrong in a way, and so it's a bit like that where it's, and I suppose it is in a way a bit naive to think that one gene controls one individual process, and it's as simple as that, because it must be. It makes sense that it's a combination of things, and so it's not just as easy as just identifying one particular gene for each thing.
Yes. So just to slightly focus back to spores, just because I can never be too long without talking about spores, because otherwise I will explode. When we talk, I'm just to
this is
maybe
what your SS has been doing to you, honestly.
Yes. To go a little bit through the kind of talk, we're talking about stage 1 and stage to help you maybe picture what's going on when I say it forms a spore. It's maybe useful to kind of have an idea of what's actually going on there. So you've got a normal cell and it's starving. It's really hot, or it's really cold, or it's really dry, and will replicate all of its DNA, just like it would for replication, and it lines them up. So you've got like a pill shape, and it's like you can imagine all of the DNA stretched out, and the difference between forming a spore and forming asexual reproduction, so forming two healthy cells, is the fact that the division is asymmetrical. So you're forming a division site that's kind of shifted to the side. So you've got a large cell, almost like forming a large cell and a small cell. And then what we might call stage 3, you've got the formation of what's called a septum. So like a line forming down the cell. The thing controlling it, the next steps, is the small cell should be engulfed by the big cell, but it can't do that when we delete the next step.
Is this what you were, you showed me those pictures of like the things and they were like attached to each other. Is this what that is?
Yes.
Okay. Yeah. So as you mentioned, there's during the sporing process, the sporulation process, the sporing process, it divides into a bigger cell and a smaller cell. and that
Yes.
comes from replicating its DNA, but not but the replication is not exactly similar to the same one like you might get in just normal reproduction of cells.
Yes.
And then it's meant to, at some later stage, combine back together, but they can't do that because you've deleted this certain gene, and that's what stops the sporulation process, essentially.
Well, not necessarily combine back together. It's almost like forming a mother and a child. So
Okay.
you have, because it's asymmetrical, you've got the big cell and the small cell. Now, the DNA lines up along, and then the big cell and the small cell split happens, and that means you've got some of the DNA in the small cell, most of the DNA in the big cell, and then you have to move. There's two sets of DNA because it's replicated. You've got to put one set of DNA inside the small cell, and then the big cell provides energy for the processes happening inside the small cell. The big cell doesn't make it in the process. The small cell is called the forespore, and that's what forms the spore, and the big cell is called the mother cell, and it kind of helps the forespore form a spore and then just dies.
Does this small cell have, because I know you said it reproduces its DNA asymmetrically, so does that mean that the DNA that the I guess the child cell, as we call it, or like the smaller cell has, is it exactly the same as the mother cell?
Yes, but when it first splits, only 30% of the DNA is in the small section.
Right.
So you've got the mother cell. The mother cell will have 70%. It's got 1.7 DNAs, and the small one has got 0.3 DNA.
Yes. Yeah, that makes sense.
So yeah, you've got that. One of the first steps, once the line forms, you've got the 30% that's in the forespore can do what it has instructions for, and then the rest of the DNA, which is currently kind of trapped within the line, has to be pushed into the small cell. And they have the same DNA. Once the kind of translocation, as it's called, of DNA happens, there's the same information in both cells, but they are differently regulated. So all the instructions are there, but only some of them are turned on. So you can only do some of the processes. So, for example, you need to be able to do one process in the small one in order to form a spore, but also you need the big cell to do something else in order to form, well, to engulf the small cell. It has to completely remodel its membrane, so it's kind of like the outside has to curve around the smaller cell and then move it inside the bigger cell, and then it has to help form chemicals and proteins that will kind of act as a coating around the smaller cell and then just die and leave the smaller cell with its really strong walls.
Yeah. Okay. So, well, I suppose it makes sense that the bigger cell and the smaller cell would have different genes turned on and off, as I suppose people would typically say, because at the end of the day, they're serving different purposes. Like the smaller cell is going to be the fore cell which forms a spore, and then as you just mentioned, the mother cell is really only there to serve the smaller cell and make sure that it completes the sporulation process. So it just makes sense that I guess some genes would be turned on and off. I suppose that makes sense, really. I think so. Does when you cut off as we kept mentioning this stage 3 where the mutants don't proceed any further, is all of the DNA transferred across by that point, or is it not? Do you cut them off while they still have this kind of 30%, 170% split in a way?
No. The point that we cut off is we prevent engulfment. So by this time, and basically, the small cell and the big cell have all the stuff they're supposed to in the right places, but the small cell can't be eaten by the big cell because the set of genes that are needed for that remodeling of the membrane to stretch around the outside can't happen because we've deleted the on button.
Right. So
as it were.
So it has all the same. So it has all the same DNA, but the mother cell can't wrap around and form this kind of like hardened wall essentially around the smaller cell, because the gene that tells it to do that is the one that's been deleted essentially.
Yeah, so it's the fact that it's like a cascade. So you've got that we've done all the steps that help split. We've done all the steps that help move from one place to another. The formation of the septum is the kind of, it's formed a line and it's moved the DNA to the right place, and it's started to make there's a lot of the word remodeling kind of could be used very generally to describe. It's just sort of because it has an entirely different metabolism. Once it's in the spore form, it has completely different regulatory processes. It uses different materials to form its coat and things like that. It's all of the steps that are involved in forming these different shapes. It's doing all of them till the point where it should be able to be eaten by the big cell. Yeah, engulfed. But we've removed that. It's not resilient at this point. It's got its DNA. The forespore is a little cell containing all of the DNA that it should have, but it's not resistant to anything because its wall has not been, after engulfment is where you get the special proteins in the wall, and you get the coat proteins, and you get dehydration and things that help it be resilient. None of that's happened yet. You've just put DNA in a small cell, which is why it should be able to reverse if it can. Because
I see. Yes.
It's just a small cell filled with DNA. So it's got all the instructions to be a normal cell, and it's got the right wall to be a normal cell. So it can just start growing if it's not. But between step 2 and step 3, there's something there's the actual thing that stops it from turning around.
So basically, because I know we mentioned before when we were going through this process that there needs to be some mutation that happens for this kind of smaller cell to be able to turn around. And yes. I guess because, so as you mentioned, when the DNA is duplicated, it is the same DNA, but some different genes are turned on and off compared to the mother cell, which again makes sense because if we were to talk about this normal process, they are serving two different purposes essentially. So is the mutation that needs to happen that the DNA in the smaller cell, it has to turn some certain genes on and certain genes off so that it basically reverses back to what a normal cell should be essentially? Well, a little bit more complicated
as
than that.
the current state of me talking to you about this, commitment has not been solved. We don't know. Okay. But we know that there are some deletions that can make things partially uncommitted. So you can quantify how good at turning around they are, and we can make them a little bit good at turning around, like 10% good at turning around. We are nowhere near the point that we should be.
Okay.
But this is because, again, it's not just a single gene. It's a combination. Yes. There's the combination of multiple genes that work together to turn things around. We gotta find as many of them as possible and then put them in the same mutant to see if that combination is what reverses it.
When you say 10% turning around, does that mean that 10% or 10% effective at turning around? Does that mean that 10% of all of the cells turn around? Is that how you would quantify that thing?
Again, there are different ways that we can turn. There are ways that we can decide quantification systems for how to consider commitment. So we measure commitment in 3 different ways that I will probably go into in a later episode because it involves some quite hefty explanations of gene regulation that I think would probably be a little bit of.
Yeah, that's fine.
No, it's all good. But we are able to quantify how committed a cell is by basically how well it's called outgrowth. For example, the small cell might be able to grow bigger again, but not replicate, or it might be able to survive and replicate, but not remove itself from the big cell. So there are because of the fact that we're getting sort of some of the genes that it needs, but not all of them, it can sometimes do, these mutants can do some of the outgrowth steps, but not all of them. And you're not getting just healthy vegetative cells, so the normal cells, when outgrowth happens, you're not just getting, oh, everything's back to normal, we're all just growing. You're getting sort of maybe defective cells, or cells that are still attached to their mothers, or cells that are a bit smaller than they should do, or cells with loads of division sites and things like that, because of the fact that it's not outgrowing naturally. We're trying to reverse engineer the process to understand what stops the process.
Yes. So in a way, it's like this, again, we'll go into this in a later episode, but this kind of measurement of how much it's able to reverse this or uncommit itself is almost like a tick box of like, there's many different things that it has to do to basically transfer back to a normal cell. And it's just determining how many of these things it's able to do, essentially.
Yeah, in a way. We're trying to get the full set of things. Well, almost, we're trying to identify the full set of things it needs to commit to sporulation. But the only way we can do that is to remove them and see if it can uncommit. So it is.
Yes. Yeah.
But yes, because we don't want them to uncommit. We just want to understand what makes them commit. The reason we're not making these mutants to try and make them turn around. We want to understand what stops them from turning around.
Yes.
This relates back to, I guess, at the level of research that we are with trying to understand things like commitment. It's fully just we want to understand a process. But once, I guess, much further down the line, because of the fact that they are resistant to basically anything you could throw at them, and then they can just, once that condition has ended, they can just grow again. So if you potentially they're resistant to antibiotics, and then once the antibiotic course ends, they can just start growing again because they are no longer restricted. So the more we understand about the start of the formation process, there are maybe we can identify some of these genes, maybe they're targets for treatments or ways to, the more we understand about spores, the more we can understand how we might combat them. Because though we're studying, in the research that I did, we studied Bacillus, and Bacillus subtilis is a harmless bacteria. Plenty of harmful bacteria and pathogens can form spores. For example, there's a bacteria called C. difficile, which is not very nice in hospitals, and it's an intestinal pathogen. It's really quite hard to treat because of the fact that if it forms spores, you can't really remove it from someone. So the more we understand about things like that, the hope would be that we can start to apply what we learn about this model organism and its processes to harmful bacteria and help us kind of work out how to treat it, because of the fact that the more we understand about commitment, the more we understand about how to target it in our treatment of bacteria.
Yeah, I suppose because it's the genes, because it's at the end of the day, it's just genes, and it's for lack of a more in-depth knowledge at this stage, it's turning genes off and off. If you understand the process of commitment, then you can understand how to maybe undo that process, which is, as you mentioned, can be very helpful in fighting different pathogens when you don't want them to enter this sporulation process, because they're a lot harder to fight off for maybe someone who's in hospital because of these bacteria.
Yeah, well, for example, hypothetically, if you could understand commitment well, you could design something to interrupt commitment and prevent it from successfully forming a spore. And then that means that you could much more successfully treat spores with antibiotics or prevent spores forming. There's plenty of other approaches we're trying, because spores are one of a really quite key problem for treating some bacteria and clearing some pathogens. There are plenty of other ways that we're trying to understand them. For example, people are doing research into the membranes to work out whether or not we could design antibiotics that could get through them and things like that. But at the moment, we're still just working on understanding, because you can't start to design effective treatments if you don't understand what you're trying to treat. So it's a sector of research that is fully exploring, how does this work? What is this? Where does it come from? What regulates this? What do these genes do? Rather than we are trying to design an antibiotic because we don't understand enough about it. We'd be going in a bit blind.
We're in the understanding phase at the moment, rather than
Yes.
the application of that knowledge to effective treatments. How long, if we're only in the understanding phase, how long have people been, I guess, how long has this been a major field of research? Do you know?
People have known about spores for quite a long time. There are lots of the papers that I've been looking at as part of my research. I'm reading papers from the 60s, but the ones from the 60s are about a good deal of the things that we know now and don't know too much more than, which is a bit sad. They were originally, we've known about them for quite a long time. A lot of things like bacteria, they are, I guess, a long time ago they saw them, but they didn't know what they were, a long time ago. And then we've kind of found out more and more, but we're still learning very much. So, for example, in the 1960s, we worked out the stages of sporulation. And but the kind of the characterization of spores came 4 decades after the discovery of spores, as in someone noticed that there were like inside of mother cells, and then 4 decades later someone went, yeah, that's a dormant structure. And then we've known about them for a very, very long time, but we haven't lots of the understanding has been very, very slow because they're so resilient. It's hard to see what's going on, and it's such a complex process to change them that it's really hard to understand what's interacting with what. In terms of commitment, the paper that the lab that I worked with and the PhD student that I worked with, the paper that they were trying to disprove is from 2005. So, and no one has managed to kind of update in a productive way since 2005 the cells, the genes involved in commitment. We've been trying to work out the set of the kind of like these 7 genes or these 5 genes are the ones that stop commitment from happening if you delete them. We've been trying to work out the subset that does full uncommitment for a long time, and it's still going. It's quite a it seems to be, there are some things that happen very fast, but now it's sort of, there's so much, you find out a lot about spores, but it's a lot of the time people are discovering small things. Like the bacteria is very well characterized, but there are some things that we don't know about. But someone might say, oh, this unnamed protein that we knew existed, that does this, but the thing that it might do isn't always going to be something really important. It might be that it supports the function of something else. So a lot of the discoveries that we're finding now are smaller and relevant for people that care, but less.
Well, it's always going to be relevant for someone, not necessarily for the purposes of medication. But no,
but I guess it's more sense. Sort of.
You say it's slow progress, but then I think there's certainly from what we'll get into in some later episodes, like it would have taken ages to, well, as we mentioned, decades to actually get to the point that we know. Because even though you just say it's not just as simple of a case of the understanding stage and then the application phase. Like there's a lot of steps, like what we were talking about before with being able to shortcut the process. First of all, you need to know that there is a process and this thing happens. Then you need to know the different stages that happens. Then you need to know how to actually make these mutants as well, and make that process standardized. So there is a lot of stuff that is, even just the stuff that we've gone through today and the introduction that we've done before we even get onto what your research is, there's so much build up that has happened to get to this stage for, as you mentioned, an organism which is literally made to be resistant to probably people prodding around it or something like that. So even though it has been going on for a while, it probably there's a reason it's taken that long.
Yeah, no, I think that's definitely right to say. And the thing as well, this is why it's so hard to explain. This is why it's so hard to kind of explain easily a lot of the processes that I've been researching, because they're behind so many layers of people discovering things and working out things. There's so much context around the sort of thing that I'm researching.
My brain's already frazzled from one episode one. But it is really interesting. It just like if you can get through the context that comes with it as well, and it has some really important consequences potentially for healthcare and treatment, and I'm sure for many other things. So it's important.
Yeah.
I'm sure that's not what people were expecting when they clicked onto a podcast about spores. Even if they have done that, maybe they're just watching The Rest is Football. But if they have done that, then they, they're probably asking, what's the point of this? Well, there you go, John or Steve, whoever you are. It's important.
Listener Steve would like to know.
Angrily typing in the comments, what is the point of this?
Yeah.
But there is a point.
So hopefully this was a fairly I know you said frazzled, but hopefully it not too much of a jumping into the deep end for spores. I'm gonna say that I think this is a good place to stop for a introduction of spores. And hopefully, if you still care about the concepts, then there will be further information available about commitment at a later date, or at the same date, but in another episode, because that's how podcasts work.
That is where we're getting to next time.
Hopefully,
Hopefully, you're not completely and utterly spored by this episode. If you're not, then check out more. Yes, as we mentioned, at a later episode.
Yes. Thank you.
Goodbye.